Good afternoon. It's the 5th of 5 invasive species Action Week invasive lunch webinars. Thank you for joining us for today's talk. My name is Doug Johnson. I'll be one of your hosts today. And we have a crew of people here to run the webinar. This webinar series is co-organized by CALIPSI, the California Invasive Plant Council, which I am a staff member. I'm joined by Jutta Berger and Dana Morowitz from our staff. We co-organized this with the University of California's Department of Ag and Natural Resources. Sabrina Drill has been our co-organizer, and she's been with us the last 4 days, yesterday as the featured speaker. Today she will not be with us. The California Invasive Plant Council is a nonprofit organization working to contain and eradicate invasive plants in California that harm our environment and economy. Igor, do you want to say a brief word about UCANR?
The Division of Agriculture and Natural Resources is the part of the University of California that transfers findings from the university to the folks who can use them. So we run the county cooperative extension offices, and we also run the community-based programs such as 4-H, Youth Development Program, UC Master Gardener Program, Master Food Preserver Program, the Cal Naturalist Program, and the 2 federally funded nutrition education programs, CalFresh and FNet. So that's us.
Excellent. Well, that's a lot. So really the connection between the university and the community, it's a very important function. Well, let me give you a couple of quick webinar tips for making the most of your experience. Whoops. If you get stuck in full screen mode and need to do something else in your computer, use the escape key on the top left of your keyboard to reduce the screen. If the box on the top right with our names or pictures on them gets in the way of something you want to see on the screen, you can grab that and move it over. You can also click, there are a few icons typically visible on top of it that allow you to minimize that view. And then finally, at the bottom of your screen, if you hover with your cursor, you should see a toolbar. It may not have as many choices as are shown on this slide, but there are 2 main choices to know about. One is the Q&A, where you can type in a question for Igor on today's topic. The other is the chat function, where you can chat and say hi to people, but you can also ask any technical questions. So if you're having trouble with audio or video, please put it in there and Dana will help you out. There is a phone-in option. We've had a few people that have had problems with their computer audio, and it helps to call in on the phone and listen on the phone. So you can listen on the phone and watch on the computer. Okay. So just as a quick sense of what we've had this week, you may have been able to participate in some of these previous webinars, or maybe you haven't. Maybe today's your first one. But we've had a lot of interesting topics this week, varying from Play Clean Go, a campaign that started in Minnesota and has spread throughout the country on how to not spread invasive species when you recreate. On Tuesday, we had presenters from Mexico who work on islands both on the Caribbean side and on the Pacific side and in the Gulf of Cortez to eradicate invasive species, and how they use detector dogs to prevent invasive species such as rats from getting on those islands. On Wednesday, we had Dr. Rachel Meyer from UC Santa Cruz talking about how to use environmental DNA to ascertain which species live in a certain water body or in a certain area where you've taken a soil sample. And yesterday, we had an interesting presentation on restoring the Los Angeles River watershed and what that looks like, from protecting native steelhead fisheries to protecting the fish that live there now in the warm waters and that the community can access. So today we have another fascinating topic, and we're thrilled to have Igor Lachan here to talk about Phytophthora and soil pathogens. All of these talks will be, have been recorded and will be posted on the same website where you registered for this webinar, The easiest way to find that website, it's a University of California website, is to Google invasive lunch, and that's the first thing that pops up. So we had a poll come up that hopefully each of you have had a chance to answer. I'm going to end the polling now. It's asking, where do you live and how familiar are you with the issue of invasive species? And I'm going to share the results. You can see that we have 94% of our attendees are from California, but we do have, 4 folks from other states and 2 people from outside the US. So thank you very much for joining us. And then in terms of familiarity with the invasive species issue, we have less than half the people who are very familiar. And that's actually great. We are aiming with this webinar series to get the word out to the public about an issue that is critical for protecting our biodiversity as well as the natural resources we depend on. I'm going to stop sharing those results. And without further ado, head into today's talk. So my colleague Jutta Berger is going to introduce our speaker.
Thank you, Doug. Well, it's my pleasure to introduce Dr. Igor Lachan from UC Cooperative Extension, as you heard. He is an extension advisor for the San Francisco Bay Area. He specializes in urban forestry, trees in our cities. Igor works on emerging issues and landscapes in urban environments, and that includes a whole host of things such as using stormwater to improve tree growth, spreading the word about new tree pathogens, and also studying other factors that can damage urban trees and the people that walk under them. He produces extension and outreach materials from ongoing research, from university research for arborists, for landscapers, and for the general public. Produces great resources. Igor hails originally from Croatia, but is academically locally grown at UC Berkeley, where he received his bachelor's in ecology, his master's in aquatic ecology, and his PhD in urban forestry. And he would like to say, go Bears. Thank you. And you're on, Igor.
Okay.
All right, I'll stop sharing and there you go. You can see your screen.
Okay. Terrific. Well, welcome one and all. Doug, can you throw up the initial poll, or did we already do that for Phytophthora?
I will do that.
Okay. So today we're going to talk about Phytophthora, and this is what Phytophthora looks like, or at least this is what how folks in Australia think of Phytophthora. So if you have a moment, how would you rate your understanding of Phytophthora? And we'll just take a few brief moments, your own understanding of Phytophthora. Not the ability to spell it, just the understanding.
Igor, are we seeing bullet holes in this sign?
We are seeing bullet holes. It's Australia, so, you know.
Got it.
Yeah. Maybe they were trying to shoot the Phytophthora. I mean, it's a very unfriendly looking Phytophthora there, just saying.
Is that a biological likeness?
Yeah, absolutely. Absolutely. It's one of the better ones I've been able to find. That's why I have it here.
Okay, shall I end polling?
Please, end the polling. So, thank you. Thank you all for participating in the poll. Oh, okay, moderate. All right. Hey, a few people know it better than me. Cool. All right. Well, you guys can correct me. So, all of the good slides in this presentation come from Professor Laura Sims and Dr. Ted Zwicky, and I want to thank both of them because without them, the presentation wouldn't have happened. And just to kind of continue in that vein, the resources that I think we've put into the chat box, I think are really quite valuable. So this is what you would want to write down. And again, the presentation is recorded. This is partially why I'm including this slide. But one of the things I want to point out is a summary that Dr. Swicky prepared about Phytophthora, and I'll go through that now because I think it sets up a very good framework for what we're going to be discussing. So when we think about Phytophthora, these are some of the more common but also less recognized plant pathogens. And they present threats to all kinds of landscapes, from cultivated and agricultural settings to the native landscapes. They can be a little difficult to diagnose because they're not very flashy, and partially as a result of that, they have been repeatedly introduced in our landscapes via the contaminated planting stock. Once they're in the landscape, they can spread further, and that is in fact what has happened. We can do something about this. We can prevent Phytophthora infestations by growing clean nursery stock and then following handling pest management practices as we go in. Eradication is difficult. There is a way to eradicate Phytophthora, especially from soil using heat, but chemical tools are not so great. There's a specific situation for sudden oak death where chemicals can be used to prevent infection, but really they're not very good at eradicating the pathogen once it's there. And we'll talk very briefly at the end about testing because it's very interesting, I think. And it's certainly very useful to see whether your best management practices are working. So it's a way of confirming that what you're doing is working, but it is not really a way to test. There's not really a way to test every single plant out there. We really have to rely on having good practices. So that's pretty much it. The outline, we'll talk briefly about the biology of Phytophthora for those of you who may be new to the entire issue. We'll then illustrate some of the specific things about this pathogen with 3 examples from California, what I call the horticultural Phytophthora, sudden oak death, and the new Phytophthora species. And then we'll briefly touch on the management. So, Phytophthora, one of the most important plant pathogens anywhere. And this is a large group of pathogens that can affect a very large swath of plants, from small, tiny plants like this one here to very large trees. And of course, if you have any connection with Ireland and you're in the US, one of the most important historical famines was, at least in part, precipitated by a Phytophthora. This is late blight of potato, which precipitated the Irish potato famine. So Phytophthora have changed history, and they continue to change our landscapes and ecosystems. So it's I think, important to understand them a bit. So let's start with the biology of Phytophthora. There's somewhere around 120 described species. We think there's probably somewhere around 500 that are out there. And they're not very flashy. They kind of look like a fungus. They make spores, they have hyphae, they will grow in culture. But the thing is, they're totally unrelated to fungi. And this is cool if you're a biologist. It's kind of like looking at a dolphin and looking at a shark. They're really not related, except Phytophthora and fungi. It's kind of like looking at a dolphin and looking at a fern or something. They're really not related at all. They just kind of look alike. But again, it's a fungus-like organism. It kind of behaves like a fungus. It'll grow hyphae, that's how it grows. It reproduces by spores, and it has multiple kinds of spores. Now, the reason this is important is that some of these are resistant to desiccation. So some of them can be used, the Phytophthora can use some of these to essentially overcome difficult environmental conditions. But it uses other spores to disperse and infect plants. And these are especially these zoospores. You see this? It has 2 little whips, 2 flagella. It uses one to actually kind of pull itself through water, and it uses the other one to steer like a rudder. It's really cool. And here's what that looks like. Let's see, these videos work roughly 1 in 10 times, but maybe, oh, look at that, it's working, wonderful. So you can see the little zoospores coming out of the sporangium, they're swimming around, and they're looking for plants to infect. And this is sort of unique to Phytophthora, having these swimming spores with 2 little whips on the sides. That's kind of cool. So how does the infection work? This is one of those dreaded infection cycle graphs, but bear with me because this has important implications for how these diseases work. So if we start with infected roots right here, these are infected plant roots, if we're in a wet habitat or a wet part of the year, then what we do is we simply make additional sporangia and additional zoospores. These are the little swimming guys that then go in and infect new roots. So it's a fairly direct way of infection. If we are in a dry habitat or dry time of the year, then what happens is we will make these chlamydospores, which are the resting stages, or oospores, again, another resting stage that can then form sporangia. Then when the conditions improve, namely when it gets wetter, they will again form sporangia, make zoospores, and the infection cycle continues. So they're able to tolerate dry conditions, but they really need water to reproduce and to infect. This is why phytophthora are sometimes called water molds, or the group of organisms, the oomycetes, are called water molds because they are associated with water. And you can actually see this very nicely. This is a infected California bay laurel, and you can see the infected part of the leaf is at the very tip where water drips off. So that's the part of the leaf that becomes infected. And water is important for the spread of this disease. So when you're looking at, and this works both at a large landscape scale and at a small scale of, say, an infected nursery. So when you look at the landscape, you see certainly, oh, woohoo, okay, okie dokie. Certainly the disease can move from one infected tree to the next, but it can also move with infected water or soil. So you can see it moving down the slope here, and you can see it moving with along a water course here. So water, and we'll keep coming back to this. Water is very important for the spread of this disease. What does the disease actually look like in a plant? Well, again, it's not very flashy. It's a typical antagonistic symbiosis, which really means it's a parasite. It comes in and causes disease. And there's really three ways this disease expresses itself. Most of Phytophthora species cause a root rot. So they decay the fine absorbing roots, and we'll see many pictures of plants that basically have no fine roots left because Phytophthora has eaten away all of them. A few Phytophthora species cause a foliar disease. They cause a foliar blight. So a close relative of Phytophthora that's super famous is downy mildew. Maybe you've heard of it. It almost put the French wine industry out of business in the mid-19th century. And that's a typical foliar blight, and I'll show you some pictures of that. And a few Phytophthora have the ability to cause trunk cankers or lesions, and these tend to be fairly fatal. If they go all the way around the tree, if they girdle the tree, those end up being fatal. Some Phytophthora then have the ability to feed on the dead tissue, so they are saprophytes as well as pathogens. And there's a few plant pathogens that work this way, and they tend to be difficult because they can kill a plant and then still persist, which a lot of plant pathogens can't do. They really need living tissue, but some Phytophthora can do this. So let's look at three specific examples, which I think will illustrate some of these aspects of Phytophthora. And the three examples are Phytophthora cinnamomi and its friends, and I'm terming this horticultural problems. This is the kind of thing you would see in cultivated yards. And when you look for the information, you would call this root rot or crown rot Phytophthora. The second example will be Phytophthora ramorum, sudden oak death. And what's interesting about that is that it's a single species of Phytophthora that causes that disease. So it's a little bit different than some of these other ones. And then the third example will be some of the new species of Phytophthora. Not only are the species new, but they're also showing up in a new setting. They're showing up in restoration plant nurseries. So I'm kind of making this distinction, but I want to remind you that Phytophthora don't see this as a real distinction. If you have a susceptible plant and you have the pathogen, you have the Phytophthora present, and the environment is right, which means typically there's some water, you are going to end up with a problem. Phytophthora is not going to say, oh, I'm sorry, I'm a wildland species and this is a commercial nursery, so I'm not going to go in. I'm simply using this division as a way of organizing things. So just kind of keep that in mind. But if we start with Phytophthora cinnamomi and some of the related ones, this is what's often called root rot or crown rot. And these are very non-distinct symptoms. The plant essentially just sort of wilts and declines. And if you dig it up, you see that it has no fine roots left. And this is frequently seen in residential yards, but it's also seen in Christmas tree farms, for example. And here you can really see in this image that movement. So the infection started somewhere here, and then with the runoff, you can see how the infection spreads as trees die. It's a really nice example of how Phytophthora moves with the runoff. What do you see on the plant when this is actually happening? Well, the thing is, you don't see much. This is what I mean about these diseases not being very flashy. Here is a coast live oak infected by Phytophthora cinnamomi, and you can see thinning of the canopy. That's one of the early signs. Then if you look further down on the tree, you see some bleeding points. Again, not very dramatic, I would argue, but just this diffuse bleeding. And what's going on? This is a good indication that there's something going on under the bark. And what's going on under the bark is this canker, which is this right here. This black dark line is where Phytophthora is actively destroying the new tissues. And you can see the destroyed tissue, the canker itself is back here, and the healthy tissue is here. And the infection front is proceeding like so. If that infection front comes all the way around the tree, it will probably lead to the death of the tree. What it's doing is it's killing the cambium. It's killing the actively dividing cells that are just under the bark, and then it will move into the xylem as well, into the tree's plumbing. Again, unfortunately, not very flashy. Now, I will say that sudden oak death symptoms look pretty much exactly like this. So this is where you really need a molecular test to tell you what it is you're looking at. What's one easy way to perhaps tell whether you have Phytophthora cinnamomi or one of his friends or sudden oak death is to look at the tree species that's affected. So here we have English oak and we have cork oak, and you can see this profuse bleeding. And on cork oak, it's a little harder to see, but you definitely see the bleeding points. This is not sudden oak death because neither of these two species is particularly susceptible to sudden oak death, but the symptoms really are kind of the same. So this is what you see in a lot of cultivated urban yards and landscapes. But remember, Phytophthora doesn't really care, and if conditions are right, this is a state park, it can affect wildland trees just as successful. So that's Phytophthora cinnamomi. Now, starting in about mid-1990s, we got a new Phytophthora, and that's Phytophthora ramorum, and this is the cause of sudden oak death. Now, if you want to know about sudden oak death, all you need to do is go to suddenoakdeath.org. That's the California Oak Mortality Task Force, and they have a great website that includes information for homeowners, information for land managers. It's just outstanding. They've been at this for over 20 years now, so they've really put a lot of information on there. The key thing to realize about Phytophthora ramorum, or sudden oak death, is that it really kind of is two different diseases. It's the same pathogen, but depending on the host, it can express as two very different diseases. On most susceptible plants, what you're going to end up with is just a foliar disease. Remember that foliar blight thing? So look here, this is, I think, either a rhododendron or camellia, but this is what you end up with, destruction of the leaf tissue. This is a redwood. They are not very often infected, but when they are, all that happens is some leaf and maybe some twig blight. So typically, this is not overly serious, although it can actually damage and kill some of the younger, smaller rhododendrons especially. But this is the key to why sudden oak death has been so successful or so devastating, depending on your perspective, because some plants will get this foliar disease and they will serve to spread sudden oak death, but not die themselves. Whereas once the disease hits oaks, and specifically tanbark oak in California and coast live oak, those have been the two primary victims. Now, oaks in the red oak group are generally thought to be susceptible, but really where we see this disease is coast live oak and tanbark oak. And these trees get a trunk canker, just like I showed you a moment ago. So as you start with this diffuse bleeding on trunk, and if you scrape the bark off, this is what you find. You find this canker, and you can actually see these cankers coming together. And once they completely circle the tree, that's it pretty much. Now, I will say that we've learned that some trees are essentially able to sort of outgrow these cankers. They're not really able to eradicate them, but in some cases, the tree is essentially able to stop the growth of the canker and just keeps growing itself and kind of overcomes the problem. But this is how sudden oak death kills trees. And the reason it's been so successful is that one of its foliar hosts, so a plant that gets a foliar disease and doesn't die, in fact, doesn't really even care much about it, is the California bay laurel. And here you have a California bay laurel leaf. Right there, that dark line is where Phytophthora is reproducing, shedding spores. And, you know, if you're a gardener, you see this, you won't even worry about it. You'll say, oh, it just looks a little unhappy. But that bay laurel is shedding those spores. If those spores hit an oak, especially a coast live oak or a tanbark oak, they have a good chance of causing disease and then killing the oak. And what's really sort of interesting is that oaks are a dead end host. So a coast live oak that is dying of sudden oak death is not spreading the disease anywhere else. Tanbark oaks will spread the disease, but coast live oaks will not. So it's kind of, I don't know, ironic is the word, but it's this is what's really allowed the fact that we have coast live oaks, tanbark oaks, and bay laurels all in the same ecosystem has really allowed this disease to take off. And it has really reshaped some of the Northern California coastal forests in the last 25 years. Disease is present all the way from Southern Oregon to about Big Sur. Although I will say, if you have those three species, you're just waiting. So coast live oak, tanbark oak, and California bay laurel, you're really just waiting for the Phytophthora to come in. There's not much of a reason why it eventually wouldn't come and do the same thing it's done here in Northern California elsewhere. And it's not only do you have the death of these large, massive trees, and by the way, larger trees, unfortunately, are more susceptible to infection. It's very ironic. And this is sort of what happens. You really end up with a refashioned ecosystem and you get a replacement, especially of tanbark oaks, with other species. So it's really changed some of our coastal ecosystems. And then about 10 years ago, something interesting happened. Now, for some of you, you're probably sitting here going, oh, Igor, who cares? I like chaparral. I don't like forests. I like chaparral. This is not that interesting to me. Well, I got something for you too. Because about 10 years ago, this is what we started seeing, maybe a little less than 10 years ago. We started seeing these native plants. This is a sticky monkey flower that were sort of failing. They were kind of dying. And when they were tested, they found a whole bunch of new species of Phytophthora in those plants. And people said, wait, what happened here? And in fact, what happened was actually fairly easy to figure out. What happened was that these plants had been outplanted. They were grown in the nursery, and they were planted out into a native restoration site. And here you've got, you know, that cycle of infection. This is just Laura's little sketch of the cycle of infection. You've got the plants being brought into the nursery. In the nursery, through poor nursery conditions, you have the spread of Phytophthora, and then they move from one root to the next. And then they're planted back out into the field. And what's interesting, or horrifying, or both, is here's an example of what is a coffee berry that's infected by one of these Phytophthora I had never heard about. And which of these two plants is infected? It's a trick question. They're both infected, except you would never tell, right? This plant on the right has obviously very few roots, and you would say, yeah, it doesn't look good, it doesn't have very many roots, something's wrong. But this plant still has roots. And what's really going on is that a lot of these native California plants are drought tolerant, which means that once their roots are destroyed, they're still able to sort of hang on and look reasonably good. Now, this doesn't look great. This is a toyon that really doesn't look very good. But in the earlier stages of infection, they're firmly infected, the roots are mostly gone, the plant still looks okay, and it gets outplanted. Remember that coffee berry? This is what happens. The next thing you know, not only is that plant dead, but now you've infected the site, and Phytophthora is spreading through your site. And this is what happens. Again, this is whole bunch of species, by the way, multiple new Phytophthora species. And you don't even need new species. I just like these new species. But here's an example from Amador County. And I really want you all to look at these two slides, because neither of these sites is very wet. This is coastal California here, but this isn't. This is inland. This is dry. And you can see how this is Ion manzanita, how the Phytophthora has just bulldozed its way through this stand of manzanita. Again, it was introduced there in a native plant liner. And when the nurseries were tested, this was a few years ago, in one nursery they found almost 50% of the plants were infected. Oof. And certainly this was due in part to poor nursery conditions. So it is absolutely true that good nursery conditions probably would have prevented some of this, but it's also true that nobody was really thinking about it because, again, native plants were not really considered as being a major source of Phytophthora. For Southern California folks, this was a recent study by Dr. Ted Zwicky, and here they are testing plants for Phytophthora, and we'll talk about that in a minute. And they detected a whole bunch of them. So the situation in Southern California is really not any better. It just hasn't been studied as much. So we had our usual horticultural situations. We have sudden oak death, and then we have these new Phytophthora in native plants. So let's just finish up by talking a little bit about management. Well, prevention, as you might imagine by now, is the key to control. So if you are growing nursery plants, the key thing is to start clean. You start with clean planting stock, and then you follow the best management practices in the nurseries to keep that stock clean. And there's some of these practices are on here, and I'm not going to read this. I'm kind of leaving this for reference. But you really, you want your plants to be clean, and you want your soil to be clean, and you want everything else to be clean. And that's about it. You're not relying on chemicals to try to suppress things. And here's just some of those nursery best management practices. Again, you use water from a known source, not just a kind of well water or some puddle that you happen to have for free. You are sure that you don't have drainage problems because, again, Phytophthora needs water to spread. You're disinfecting your growing areas, your pots, your tools, and so on. And you've trained all your staff and your interns so that they know to look for plants that don't look so good and then separate those plants and really keep records of what's happening. Once all of these BMPs have been implemented, and I really want to stress this, most of the native plant restoration nurseries have been trying to implement these. Once they're implemented, you really have a huge decrease in incidence. The Phytophthora in this one nursery, in these 5 nurseries, and these are small nurseries, they're non-commercial, these are kind of backyard nurseries almost. The Phytophthora incidence went from 15% to 3% to nothing, 0% in 2 years. So it's really possible to move the needle, as they say, by implementing best management practices. And one of those best management practices is testing. And I again wanted to show this slide from Dr. Zwicky because it's cool. This is how plants are tested. So you have your usual plants up here, and all they do is pour water through the liners, just like you would with irrigating the plants. And the water is caught down here, goes into this special bucket and drains out. And you see this green thing here? That's a pear. It's a green pear. Because remember those little swimming zoospores? Yeah? They will swim up and infect the little pear. And all you do is you incubate this bucket of water for a few days, and you pull out the pear and you look for brown spots. And if you see a brown spot, you cut it out and then you culture it. And this is a way to bait Phytophthora out of the soil. You use the pear as a bait because otherwise it's actually kind of difficult to just look at soil and isolate Phytophthora. So this is super cool. Now, just a reminder, it means not using sick plants, not keeping sick plants, and not buying sick plants even though they're on sale, which is invariably what I do because they're on sale. If you're buying plants, and this goes for, I think, everybody from home gardeners to landscape managers, if you're not absolutely 100% sure about their origin, about their handling, can you quarantine them for a few weeks? And if you do this, you're basically giving the disease a chance to get expressed. And then you didn't outplant them, right? You didn't put them into your landscape. They're still in the pot. And of course, you put them in the lowest spot in your landscape. So any water that comes through is not draining into the rest of your landscape, right? This is why this is set up like this. So can you quarantine your plants? And of course, once you've planted them, are you then checking on them to see if they suddenly start looking not so great? And again, this is a good indication there's no roots left. And there's really not very many plant pathogens that will do this. So this is a good indication that something bad is going on. So that's sort of the plant side. Containers and soil, you can disinfect. Turns out the Phytophthora is actually quite easily killed by steam heat. So if you're able to pasteurize soil, you can do this, and you don't have to, you're not sterilizing the soil, right? So if you look at this chart here, weed seeds, you really have to kind of bake the soil. But for Phytophthora, 50, 60 degrees Celsius, and Phytophthora is gone. So this has been very successful in some places, this method of sanitizing soil so you can reuse it. And then the final thing to do is to just sanitize your tools, your trucks, your shoes. This is not the most efficient way of transmitting Phytophthora, but it absolutely happens, especially if you have large construction machinery on restoration sites and you have, many pounds of soil that is now stuck to the tracks or the tires and so on. Then yes, you can actually transport that infected soil from one site to the next. And if you're doing work during the rainy weather, that's perfect for Phytophthora, right? So you really want to just wash those machines on site, and your boots and your equipment, just so to not spread things between sites. So I think that's about it. And I just want to sort of run through the summary again, and we can talk a little bit more about some of these things. But I hope I've convinced you that Phytophthora of various kinds are a massive threat, both to horticulture, to agriculture, and to native habitats. Diagnosis can be difficult, but it's not too difficult to spot the symptoms if you know what you're looking for. If you know you're looking for those foliar blights, for crown thinning, for bleeding on trunk. And then the question is, okay, well, how do we not get it in here? And that goes right back to nursery practices. Did we grow this stock in clean conditions? Did we start with clean stock to begin with? By the way, it's perfectly fine to use seeds for restoration, and few people do, and we can have a whole discussion of why that is, but that would be one easy method of just bypassing this entire Phytophthora business. Did you follow the sanitary working practices in the nursery, and then as you outplanted the plants? If you need to use heat eradication, again, they're trying this in a number of sites. It works, but it's difficult, as you might imagine. Bringing a steam generator out to a restoration site is not the easiest thing to do. Chemical suppression, just a few words. There are fungicides that will suppress Phytophthora. Ironically, the very first fungicide, which was the Bordeaux mixture, the copper sulfur, that was developed for downy mildew, which is relative to Phytophthora. So very, very ironic. But the thing is, fungicides don't work very well on Phytophthora. They can be used to suppress Phytophthora populations that then may prevent infection. This is sometimes done in the nurseries. But if the plant is already infected, all that this treatment is going to do is going to suppress symptoms. So then when you take the plant and you stop dosing it with fungicide and you plant it, all of a sudden it has symptoms. That's why I'm saying, if you ever have any doubts about your plants, just quarantine them for a few weeks and they'll start to express symptoms, then you know not to plant them. The one special case or exception is the use of phosphonates for prevention of sudden oak death infection. And it's a specific case where trees that have not been infected with sudden oak deaths can be sprayed or injected with phosphonate, with phosphorus acid salt, to prevent infection by sudden oak death. This does work, but it has to be repeated. So if it's something you're interested in, the information is on the suddenoakdeath.org website. Again, it does work, but it takes some work to keep going. And it's for things like native plant restoration, this is really not an option. We need to just have clean plants. And then a lot of folks have asked about testing. testing is great because it tells you whether your management practices are good, right? Testing tells you whether what you're doing is helping you produce clean plants, but you're not going to test every plant. And it's just, and this is Ted's way of framing this. Again, Dr. Ted Sweeky of Phytosphere Research. We don't test every steak that you buy at the supermarket or every bunch of broccoli. We test a few of them to make sure that the food handling system is set up correctly and that the food is being handled safely, but we're really relying on the system to keep us safe, not on testing every individual thing we decide to eat. So I think that's about it. I just want to thank, again, Laura Sims. Many of these slides are from Professor Sims, who used to work in Matteo Garbolotta's lab, and she's really one of the experts on these new Phytophthora. And then Doctors Ted Sweeky and Elizabeth Bernhardt, who are principals of Phytosphere Research, and they probably know more about Phytophthora than just about anybody in California. So thank you, Laura, Ted, and Elizabeth. And I will put up the resources slide, and I will end there. So if there are any questions, I think we have a few minutes. We can talk, or we can go back to any of the previous slides.
That sounds great, Igor. Thank you so much. Yes, if you can leave this resources slide up, I think that will help people copy anything down they want to, or maybe take a screenshot. We also intend to send out a follow-up email to thank everybody for coming, and we can put information in that. So yes, please do enter questions that you may have into the Q&A, and Yuta will share some of those. I'm seeing that it's about 1:45. We will continue answering questions as long as there are questions, and Dr. Lachan has time and energy to field them. But feel free to leave when you need to, of course. You will, when you leave the webinar, see a brief survey just asking you about your experience and ways we can make this better. Please do fill that out. It helps us maintain this webinar series. Saying that, Igor, it reminds me of the second follow-up poll.
Ooh, yeah, follow-up poll.
Okay.
Put up the follow-up poll, or we can answer questions and then put up the follow-up poll, whatever you prefer.
I'm gonna throw this up just to gauge
there we go. Bam.
how you're thinking about Phytophthora. Do you feel like you know a little bit more now?
All right, I think I'll step in with some questions because we have 15 and counting. I'm sure there'll be quite a few more. So we had a fair number of people interested in management, of course, and they're concerned. So first of all, in an urban environment, Mary Delaval is asking about how to control for Phytophthora in your yard if you already know that you have it from a homeowner's perspective.
Well, so a lot of it's going to be water management. Phytophthora, also people who knew things better than me at the beginning now know less. This is not good. This is just as far as changing learning. This is not.
They're impressed with how much you know. They've learned that you're an expert.
Okay, good. We'll go with that. Thank you, Doug. Thank you. Doug, I'll have you everywhere from now on. This is fantastic. So when it comes to managing Phytophthora in residential yards, it's really going to be about water management. You're not likely to eradicate the thing. Some plants tend to be more susceptible, some plants tend to be less susceptible. So you kind of want to look at your plant selection and maybe decide that some of our native plants, unfortunately, are very susceptible. So that's not ideal. But it's also true that having generally good conditions in the yard, so not having flooding, having reasonable water content in the soil, so not too high, but also not too low, so you're not constantly stressing your plant, those things tend to help. There is a little tiny itsy bit of evidence that having more organic matter in the soil is helpful in that, not because it does anything to Phytophthora itself, but because it encourages the growth of other soil organisms, some of which tend to be antagonistic to Phytophthora. There is not really an easy way to just kind of get rid of Phytophthora out of the soil. On the other hand, if you have areas that are infested and you let them go fallow for a couple of years, you do reduce the inoculum in the soil, you reduce the inoculum in just about everything. So depending on where you are sort of on the spectrum of how bad things are, it is possible to actually just replant with another set of plants and not necessarily get reinfected right away. Again, if you have waterlogging, if you have standing water, if you have sort of really unhappy conditions, then yeah, you can get reinfection right away.
Got it. On that same theme, then moving to a wildland environment, some folks were wondering about the effectiveness of fire, of controlled fire, and of solarization.
Yeah, I don't know that there's a whole lot of research on either of those. So fire is used actually to manage Phytophthora, or at least it's been tried, especially in Oregon. I don't know, to be quite honest. I'm not sure I would entirely go into recommending prescribed fire as a management tool for Phytophthora, in part because you probably have to do a lot of fuels management before you reintroduce fire. And this is definitely the case in a lot of our coastal ecosystems. The fuel load is now so high that things are going to be kind of interesting. And especially with sudden oak death, where you have a lot of dead trees because of sudden oak death, you end up with much larger fires. And this has been shown. So it sort of presents a double problem. You really have to do fuel management first, then reintroduce fire. I think there's some evidence that fire helps, but it's not like a linear relationship, and it's often not very easy to use.
Permitting involved too.
Oh yeah. I mean, all of the things that go with prescribed fire still apply, except remember, you're now dealing with more dead stuff. Yeah. So you have more fuel.
Got it. And so you also described a lot of handy best management practices that people could incorporate. How much are those being, and is disease control being incorporated into urban plantings, urban tree plantings, and by cities?
Yes and no. So I would like it to be. But I think people are still surprised when they come in, 2 or 3 months after they planted a tree and the tree's performing poorly, and it's clearly showing signs of Phytophthora. And I'm kind of wondering, well, did you hold that plant at all? Like, did you really get it from the nursery yesterday and plant it today? In a lot of cases, that's what happens. But it's very often not ideal. And certainly, look, if you have a nursery supplier and this happens, then the next tree from that same nursery supplier, I would think you would want to quarantine. You would also want to go back to the nursery and say, look, folks, this tree has Phytophthora. Are you sure it didn't have it when it came out of the nursery? What did you use? Did you use one of the chemicals to suppress, one of the fungicides that suppresses Phytophthora? And if they say, yes, we do, and a lot of nurseries do, well, then again, I would argue you'd want to quarantine those trees before you outplant them. There has been a lot of, by the way, none of the nursery stuff is new. This was known in the 50s because these basic Phytophthora diseases are not new at all. So there are some nurseries that have always produced good quality trees and good quality stock. And there are some nurseries that still haven't gotten on the bandwagon. So I think it's perfectly fine to have that conversation with the nursery and say, okay, what have you done? To the credit of native plant nurseries, a lot of them have sort of jumped on this quite quickly and have made improvements. But it's absolutely okay to have that conversation with your supplier. Where are you with this? What are you doing? Are you aware? What steps are you taking?
That's great reinforcement too. Dave Thompson is asking, wanting to go back to your comments about seed, that originally there was some concern that seed might carry Phytophthora spores. Sounds like there's not anymore, or any further comment?
I think seed is relatively easily disinfested. So even if you are concerned, there are easy ways to disinfest the seed, which is very different from seedlings. Seedlings, they're either good or, that they're not going to be used. Seed, you can typically do things to disinfest. I do understand that for a lot of folks in a lot of situations, this is actually not that easy to do, but at least in theory, it's easier. Whereas with seedlings, with plants, even in theory, it's pretty much impossible to do anything once the plant's infected.
Okay. Going back to the origins of this wonderful past sudden oak death from Asia, but specifically, is there any more knowledge about where it comes from?
I certainly don't know. My understanding is that it was introduced multiple times into California and into Europe in different places. And that's how we ended up with different mating types and different genotypes of the Phytophthora. I don't know that anyone's actually pinpointed where it came from.
Interesting.
Yeah.
All right, and Joey Durrell Canepa, I think you're going to have a few questions. They're remote diagnostic questions now. Is asking about an experience he had with Ericomeria ericoides, the golden bush in a dune restoration site that seemed to have died off pretty severely in 2019, but it wasn't tested, or it didn't come out to have Phytophthora. It was tested for that. And it also didn't seem to have insect damage. Is there a new, like if you do a Phytophthora test, are you excluding the possibility that, or including that it could be another Phytophthora that you don't know yet?
Depends on the test, but no, the basic screening test for Phytophthora will pick up all of them. And in fact, they'll pick up a few things that aren't Phytophthora. They'll pick up some Pythium as well. So no, if you're doing a basic screening test for Phytophthora, I'm not saying it doesn't have Phytophthora, the test is negative, because it's possible to get a false negative for a whole number of reasons. But no, the test itself, if it were done properly and all that, no, it would pick up an unknown species of Phytophthora just fine.
Got it. All right, and so now we have another ID question from an arborist from Southern California who has a situation where they have a large coast live oak and there were native plantings, including bay laurel, planted around it. And now the oak has cankers. Could it be that it has sudden oak death even though it doesn't occur in Southern Cal?
Well, you would have to have brought it in on something. So either on that oak, which is possible, or on the bay laurel, which is also possible. I will say that nurseries that grow oaks certainly have been reasonably attentive to the problem of Phytophthora. And I'm hoping the nursery that grew the bay laurel was attentive as well. It would be interesting to find out, but it's impossible to tell just sort of by looking at it. If, the symptoms really don't look very different. The Phytophthora ramorum versus Phytophthora cinnamomi, not too different. So you could have the tree tested, you could have those bleeding cankers tested. Now remember, that oak, if worst case scenario, it has Phytophthora ramorum, is not going to spread it anywhere. So I would focus on the bay laurel and look and see whether that bay laurel is exhibiting any symptoms. It will. So if it is infected, you will see that leaf blight on the bay laurel. And that's relatively easy to test for. So in the universe of how difficult or easy things are to test for, bay laurel is actually fairly easy to test for. So one way to do this would be to join the BioBlitz, the SOD Blitz, which Matteo Garbellotto's lab organizes every year. I don't think they've made it to Southern California, but if you wanted to volunteer, I'm sure they'd have you.
All right, and that would be including then just sending specimens?
There is a specific procedure they want you to follow. So if you go to the SOD Blitz website, you'll see what's involved. The Garbellotto lab does offer a testing protocol for arborists, and they have on their website, the entire protocol is there. If you go to suddenoakdeath.org, I think it's linked over there. And if not, just send me an email and I'll send you the links.
Got it. So Elizabeth Mesquita is asking whether Phytophthora can be present in indoor plants. And
yeah.
Okay.
Yeah, it can. Absolutely. Phytophthora, different Phytophthora have different host ranges, but yeah, I'm sure we could find a Phytophthora in an indoor plant. You can find a Phytophthora, all kinds of stuff. I'm pretty sure one of my aquarium plants has it.
Oh, great. So, and you're not throwing it out.
No, it's
Okay.
What
All
do you
right.
call it? It's aesthetically interesting.
That's discretionary, yes. Okay. So, all right, I think that is answering also the questions of what to do about treating a house plant. So it's...
There's not much you're going to do. So if it's a foliar blight, so something like a downy mildew kind of thing, there are contact fungicides that would protect the new foliage. So typically they're not going to eradicate what you got, but as new leaves come up, those fungicides will protect it. But if it's a crown rot or root rot, there's, I would argue, not much to be done other than starting over. It wouldn't be my first guess for why a houseplant is declining, but it's absolutely definitely possible, yeah.
Okay, good. Do you think that Phytophthora will is able to affect blue gum eucalyptus?
I'm pretty sure there's at least some Phytophthora somewhere that's able to affect blue gum. I wouldn't be surprised if Phytophthora cinnamomi were able to do it. So Phytophthora infections in cultivated landscapes really used to be thought mostly as a matter of water management. So like if you have standing water, if you have overwatering, waterlogging, that kind of thing, you will get Phytophthora. Well, that's kind of wrong, isn't it? Because you still need the actual pathogen, right? So we've sort of changed our thinking here. You really, it's an infection. You need the pathogen, right? It's not just enough to have poorly managed yard or nursery. But if you have the pathogen and you have poorly managed yard, then it's quite possible that you're going to get infection. Some of these are going to be self-limiting, and this is true, especially for Phytophthora cinnamomi. They may infect the tree, cause some damage, and then the conditions improve, and the tree is essentially able to outgrow it. That happens, and I think it happens a lot more than we notice, but that definitely happens.
Got it. So Jess Van Brown is asking whether there's any evidence that there is resistance developing in live oaks or tan oaks, or is it just... Yeah.
That's an excellent question. I don't know. There is... it is thought, there's an entire research group at Berkeley that is looking at markers that would suggest resistance, because what they found is that there are places that are very heavily infected with sudden oak death where there are individual trees left standing. And there are two theories here. One is, well, they just haven't gotten hit, or they haven't gotten hit hard enough, okay, or something. And there's a second theory that says, no, they're actually resistant, or if not resistant, they're tolerant. In other words, they get infected, they get diseased, but they're able to outgrow the disease. I think the jury is still out. I don't know anyone who is growing live oak species that labeled as resistant. I have not seen any of that. So.
Okay, but if the infection is somewhat minor and you remove the source of tan bark, it's possible that...
I'm not sure there's much you're going to do. So if you have an infected oak, number one, you need to have it inspected by an arborist, because sudden oak death infected trees can fail very suddenly. And in fact, the whole sudden thing refers to them often failing, meaning breaking suddenly. So definitely get an arborist. But if you have an oak that's already infected, there's not much that I can think of that would really affect the disease. Now, there are things that you can do, namely cultural care, that'll affect the overall health of that oak, and it may be able to essentially outgrow the disease. Again, I think it happens more often than we think. But there's not exactly like a magic potion or a fungicide or something like that that would arrest the disease. I don't know if that makes sense. Okay.
Yeah, that does. It's not very uplifting, but yeah.
No, it's really not. But it's an important thing to realize. There are two important things here. One is there's not a curative compound that you're going to apply to and infect the tree to cure it. But the second thing is that tree, that oak, specifically coast live oak, is not going to spread the disease to anything anywhere. So if you're not concerned about that oak failing and crushing your house, your car, your cat, your mother-in-law, if it's just somewhere out there, then maybe there isn't a great rush to remove it. Conversely, if the oak is right next to your house, please get an arborist.
Got it. Yeah. All right. Carrie Olson is asking about water resources. So, for instance, rain barrel collected water. Is it possible to be inadvertently moving spores?
Phytophthora? Well, that's an interesting question. Water that's never hit the ground, I would argue, is not likely to be contaminated. I mean, anything's possible, I suppose, but you're mainly concerned about water that's hit the plants and then hit the ground. So I suppose, yeah, if you have a bay laurel that's dripping into your rain barrel, yeah. And we actually had this situation where there were large commercial compost piles that were shaded by nice, beautiful bay laurels. Not a great situation, because that's exactly what was happening. They were actually contaminating the compost. But if it's your clean roof draining into your clean rain barrel, I don't see where you would get Phytophthora contamination.
Good. And can you point folks to a resource for learning which California native plants are less susceptible to Phytophthora?
Yes. So I'm not sure you're going to exactly get lists of this, but I would start with Calphytos. That's the, if you go to Calphytos, that's the Phytophthora in native habitat restoration settings. They have information on things like this. But again, I think it's more important that you have material that is clean than something that's resistant. Resistant is kind of a relative term with Phytophthora, to be honest.
Yeah. Doug Mandel asks, how prevalent is it, like where are the hot spots in the wildlands and in ornamental nurseries, without calling any specific nurseries out? Sure.
Well, I don't know that hotspots is really the right word. There's a few restoration projects that have gotten infected. So I wouldn't necessarily worry about, like, I don't know, walking through a restoration project. I would be sort of more concerned about, and again, this maybe isn't something that's necessarily of great concern to homeowners, because you're not likely to buy plants from a wholesale restoration nursery. Remember, these nurseries really produce plants for restoration projects. Large commercial nurseries, oddly enough, have been reasonably okay with handling Phytophthora, in part because they got kind of scared over sudden oak death and some of the classic Phytophtheras. So when you're buying a plant at a garden center, I wouldn't be super concerned. I think it's still an okay idea to quarantine the plant for like 2 or 3 weeks. Just keep it in the pot, water it, and see if it still looks good. But I mean, I don't want to super scare people. I am sort of suggesting that if you have a great deal on a whole bunch of plants because they were left over somewhere, then it might be worth asking, like, well, wait a minute, where did these come from and how are they handled and all that. But if you're really just buying plants at a garden center, again, I think quarantining is a great idea, but I'm not sure you have to sort of panic about it. And definitely, the other thing that people never do, apparently, is to actually wash off the soil and look at the roots. So if there's no roots left, it's a pretty good indication that, okay, this is not what you want to plant. I think one thing that is new was this idea that California native plants were not a source of Phytophthora. Well, that's wrong, right? And I think that's the point I was trying to make with that third slide. They're not any more necessarily infected than all other plants, but they can be. That's what I'm saying.
Got it. Yeah. And let's see about just confirming for folks that Phytophthora cinnamomai is fatal in oaks.
It's fatal in some oaks. So Phytophthora cinnamomai is not the most virulent Phytophthora. It on some trees, right, some individual trees, the disease will progress to death. In others, it won't. You may end up with these bleeding cankers that keep coming back year after year, but the tree is growing fine. This is something you see occasionally. It's not necessarily a reason for panicking, but it'd be a good idea to get an arborist and kind of get a sense for how that tree is doing overall.
Mm-hmm. Got it. Just to connect with talk we had a couple days ago, Doug Mandel's asking whether one can use eDNA to diagnose Phytophthora.
I don't know. I honestly don't know. I would think that eDNA would tell you have Phytophthora, but we have Phytophthora.
Yeah.
We have a lot of Phytophthora anyway. So
yeah.
If you could do it from a specific site, I think that would be interesting. Yeah.
Yeah. Right. And it might. So you're not sure whether it would be able to tell at a species level, or.
It might be able to tell us. You probably need the right primers and all that. I think it'd be easier to actually test the plants. The other thing to realize, and this is important, is that it's actually fairly difficult to recover Phytophthora from water or from soil. So if you just take a handful of soil and you say, is there Phytophthora in here? The answer is, I don't know, but it'll almost certainly come back negative. It's just very, very inefficient. So the way to do it, this is why I showed you that green pear. The way to do it, to test for Phytophthora in soil or in water, is to bait the soil or water. You can bait the water with leaves, and the leachate that comes out of soil you can bait with green pears. But that's how they actually go about finding Phytophthora in soil and water. Otherwise, there's not like a magic test where you like dip a test strip into water and it comes up positive or negative. That's not. You can do that for plant tissue, but not for soil or water.
Mm-hmm. Got it. And for homeowners who would like to know whether they have sudden oak death or another Phytophthora, which of these resources can they go to get testing?
You will need to hire an arborist. I would start with the suddenoakdeath.org and sort of read through their materials. You'll ultimately need to hire an arborist, and they will probably need to take a sample and send it off to Matteo Garbolato's lab to be tested. I would sort of think through, before you do this, how you're going to respond. In other words, if I tell you you have sudden oak death, how's that going to make your decision different from if I told you you have Phytophthora cinnamomi? So I would kind of mentally think through that before you invest a lot of money into testing, because it may not matter. You may have a tree that's in bad shape right next to your house. You're very concerned about it failing. Does it matter if it has Phytophthora ramorum or Phytophthora cinnamomi? Conversely, you have a tree that's far out in your backyard meadow somewhere. Again, does it really matter? Just kind of think that through. And again, Sudden Oak Death has really good, the suddenoakdeath.org website has really good information.
Okay, excellent. Going back to the eDNA, I believe one of Rachel, our Wednesday speakers, lab members is participating, Wai Yin Kwang, and he or she is mentioning a website in the Q&A session here. It's Data UC eDNA about detecting Phytophthora throughout California. So that's
great. Terrific. Okay. Terrific.
Yeah. All right. Are we still good? We have a few more questions. How's your energy level here? Okay. Everyone else who is interested can stay, but don't feel pressured to.
Right.
Yeah, lots of interested folks. People are asking about mulch, whether it's safe to spread mulch from an infected area.
Well, that's a tricky question. It's a little difficult to answer. So, and it has to do with sort of your risk tolerance. The answer is probably, yeah, probably, because it's difficult to get, remember that thing how oaks don't infect other species. So coast live oak that's dying of sudden oak death is not going to infect something else. So if you grind up that, if you cut down and then grind up that oak into mulch, is the mulch going to infect someone else? Probably not. But then if you ask me, okay, so should I take that mulch from that infected oak and truck it, halfway around the state? I would argue, man, no. You would really want to use it on that site. And the logic being, well, you have Phytophthora ramorum on the site. And again, remember that Phytophthora ramorum, especially sun oak, that it's not going to infect most things, or if it does, it will cause that foliar blight, which is not very important. By and large, mulch seems to be safe, but do I have 100% certainty on that? No. And would I recommend moving it long distances? No, I wouldn't.
Got it. So we also have a question about which protocol would you recommend for arborists to follow when addressing Phytophthora ramorum possibilities?
Sednodet.org has information for arborists as well. You're going to sort of want to employ a combination of a couple of things. One is you're first going to want to make sure that the tree species is actually in the susceptible list, because I get calls all the time. My tree has Sednodet. What tree is it? Valley oak. Well, no, it doesn't have Sednodet. It may have another Phytophthora, but it doesn't have Sednodet. So it's very obvious, but it's the first thing to do. Then the thing to do is there's an app called SOD Mobile, S-O-D Mobile. It's free. And what it will do is it will try to calculate the risk at your location based on the results of these SOD blitzes. So SOD Mobile, it's the app for iPhone, Android, whatever. It doesn't work perfectly. I know it doesn't work perfectly, but it's a good start. And if it works for your location, so you'll be standing right there. You need to be standing at the location where you want the app to calculate the risk. And you'll click on the tab that says calculate risk. And it'll look for the data that have been submitted from that area and try to give you a risk rating. Beyond that, there is a way to test the bleeding cankers if you have an oak. Matteo Garbolotta's website has the details on that, or you can call me and we can talk about how to go about setting that up. It's not excessively expensive. It's, I think, $100 or $200 to set up, and that's probably $100 per tree to actually, but there is a procedure to actually you have to scrape off the bark, find that margin of the canker, take the margin samples, put it into the Petri plate right there in the field, seal the plate, and overnight it to the lab. Again, I would stress this question of how is this information going to help you? In other words, how would the knowing that you have Sednodet change your recommendation? So, but I'm happy to talk to any arborists who want to have that discussion.
Yeah. We have Rachel Dyer asking again about water. This time, is it possible to see contamination spread through well water supply and irrigation in an infested area?
I think where you would have well water concerns would be in a nursery setting. And maybe less so when you have a deep well that comes straight up onto the plants, more if you have a kind of shallow well that then gets into some kind of a holding pond, and then maybe the holding pond has a nice bay laurel shading it, that kind of thing. The well water thing is really for nurseries. Do you know where your water comes from? Is it just from the nearby creek? Because that's free. So for homeowners, I would not be that concerned. I mean, this is not, the pathogen is not sort of magically horrific in some ways. It's not, it's not a super pathogen, but it's just, if we're not thinking about it, then we can have it kind of circulate in water. And that's what has happened in a number of nurseries.
Yeah. Local.
And a number of nurseries have protocols that nurseries frequently recycle their water. That's the common thing to do, is to recycle water. And then the question is, how are you treating this recycled water? And there are slow sand systems that will take out Phytophthora. But again, it's a matter of having a system, maintaining a system, and occasionally making sure that everything's working as it should be.
Right.
Homeowners, less of a concern. I wouldn't sort of panic over that.
Got it. Yeah. And as far as the, this kind of goes back to just general, when you talk about new Phytophthora, can you kind of define that? How new is new?
Well, I don't know. That's a good question. Where's my, yeah, when you look at some of these species, you have hybrids. So some of these really are brand new species because things hybridize. And this is one of the reasons why I wanted to talk about this. It is possible to make things worse. If you are careless, it is possible to actually have a nice Phytophthora party in your nursery where species will hybridize. So how new is new? Well, these are all new to me because I had never dealt with them before. But I think what I mean by new is that they were not commonly present in nurseries earlier, and now they are. And they were not ever even thought about in native plant settings, and now they're here. So in that sense, they're new. And when you look at some of this situation like this, well, this is not a new species, right? It's Phytophthora cinnamomi, but it was introduced in this habitat where naturally it would never be there, right? It just did not occur there. And this is not just California. This happened around the world. Australia, large swaths of Australia have been, this is really how it looks. And it's not that the species is new, it's new in that habitat. So I know it's kind of a squishy answer, but that's really what it is. I mean, it's kind of almost the definition of invasive species, right? It's not the species is new, it's new in that habitat. Having said that, look at this table. Look at this table and look at some of these names. Look at that. That tells you they haven't even figured out what to call it yet. So some of these really are new species. And again, they may have been bred in that very nursery. Cool, huh?
Yeah, constantly changing. How about with global warming, with climate change? Is there some thought that Phytophthora might be suppressed some from spreading, or?
Oh, that's the most hopeful comment I've heard ever. No, absolutely not. I mean, look, and this is what I'm a little afraid of with Southern California folks, because they're looking at this going, meh, look at this. This is dry as a bone. This is Amador County. Amador County in the summer, it is dry. So no, I don't think so at all. The range may shift, but I don't think it's going to make things better in any way, shape, or form. No.
Got it. Yeah. Jane Song is mentioning that it would be great to have a list of nurseries that supply for restoration that have known safe practices. And I know some, the qualifications that people are putting out there. What can you say?
Yeah. So there is a new nursery program that was sort of piloted, AIR Association Enhanced Restoration, something like that. If you go to these, especially the Cal Phytos website, it may take you to the to some of the other websites that cover this. So there's definitely going to be a list of nurseries that are participating in that pilot program for improved nursery practices. And one of the big places that's sort of been involved in figuring out these practices is actually NORSDUC, the National Ornamental Research Site at the Dominican University of California in San Rafael. And again, if you can't find these lists, send me an email. I'm sure I can find some more information. But even more than having a list, I think you want to have a conversation with the nursery, like, what are you doing about it? And if they say something like, meh, never happened here, never worried about it, we got fungicides, go away, then maybe you want to try another nursery, right? Whereas if they engage you in a conversation, say, well, actually, we have thought about it. We now have a new protocol. We're reasonably confident that our plants are clean. Okay, that's certainly, if you are a landscape manager or you're buying plants for restoration projects, then you definitely need to have that conversation.
Got it, yeah.
And I understand, just to be totally clear, I like nurseries, I work with nurseries. It's not the easiest business in the world. But so it's less about sort of taking an accusatory tone and more about, okay, what kind of a collaborative conversation can we have about how you're approaching this problem? That's really what it is.
Got it. And many have best management practices now.
Yeah. They yes, to varying degrees, but yeah. And I'm simply saying you want to maybe avoid those who haven't even heard of this yet. But again, for homeowners, for kind of individual gardeners, this is going to be a little bit less of a problem. For land managers, restoration managers, native settings managers, it's going to be more of a problem.
Yeah. Drew Reddy is asking whether bark sprays like phosphoric acid and pentabark are still considered effective at treating or prolonging the life of a landscape tree.
They were never intended to prolong the life of an infected tree. They were always intended to prevent infection on a tree that isn't infected. Now, is that to say that they are never applied to trees that are infected? No. That's, you can buy these things yourself. You don't even need an arborist. Now, they are pesticides, and so you do need to follow the label. Please, if you buy them, please read the label, follow the label. But really, the place where efficacy has been shown, let's put it that way, let's put on our sort of scientific framework here. The setting in which efficacy has been shown is when they're applied to trees that are not infected, that are in a setting that has sudden oak death. So you have a high value oak that you're trying to protect. Yes, the phosphorus acid salts plus a surfactant if you're applying them through spray, or just phosphorus acid itself if you're applying them through injection. That has been shown to work. But again, you will be reapplying it. And if you go to suddenoakdeath.org, you will find the protocol for how to do this and how often to do it.
Excellent, yeah. All right, I think we are down to the very last question, unless another one gets wedged in here. Has horizontal gene transfer between Phytophthora and plants been detected ever?
That's very easy for me to answer because I truly don't know. So I'm not even going to guess, and I will say I really don't know.
Got it. We will wait for the science article when it happens. But it's possible. Anything's possible.
Yeah, it's
all right. Well, that's it. And the audience says thank you.
Yeah, well, thank you, audience. You were wonderful. Yeah, thank you all. And Doug and Jutta, take it away.
Right. Thank you so much, Igor. That was really informative and thrilling to hear all the information, although very sobering. Thank you all for joining us today. Please do fill out the follow-up survey when you get that. And join us next year for Invasive Species Action Week. And in the meantime, please do take any action you can to stop the spread of invasive species. Take care.